This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this deep dive into the hidden history of life, Anoxygenic Photosynthesis. Long before our planet turned blue and green, ancient microbes were already mastering the art of harvesting light without releasing oxygen. In this narration, we'll journey from the violent radiation of the Archean era to the mysterious banded iron formations and the billion-year biological stalemate that shaped our atmosphere. We will explore the infrared superpowers of these deep-dwelling alchemists and discover how their ancient metabolic secrets are now powering modern green technology. Let's begin. Chapter 1. The First Light Catchers. Long before the first leaf unfurled toward the sky or the first blade of grass broke through the soil, Earth was a world of alien chemistry and violent radiation. To understand the origins of photosynthesis, we have to look back nearly 4 billion years to the Archean era, a time when our planet would have been unrecognizable to any modern observer. The atmosphere was a thick, suffocating shroud of nitrogen, carbon dioxide, and methane, almost entirely devoid of the molecular oxygen we depend on today. Without an ozone layer to shield the surface, the sun's ultraviolet rays hammered the landscape with lethal intensity. The oceans were not the deep blue we know, but likely a murky green, saturated with dissolved minerals and volcanic gases. It was in this harsh, low-oxygen environment that life achieved one of its most significant breakthroughs, the ability to capture the energy of the sun. This was the birth of anoxygenic photosynthesis, a process that transformed the biosphere long before the evolution of the oxygen-releasing systems used by plants. Before the development of light-harvesting systems, the earliest microorganisms were likely chemolitho-autotrophs. These primitive life forms survived by scavenging chemical energy from the Earth itself, specifically from deep-sea hydrothermal vents. These vents have been a feature of the seafloor for over 4 billion years, providing a steady supply of hydrogen-based compounds through a geochemical process known as serpentinization, where seawater reacts with magnesium and iron-rich rocks in the crust. In this pre-photosynthetic world, primary production was strictly limited by the availability of these geological chemicals. Microbes would reduce carbon dioxide into organic matter using hydrogen gas as an electron donor. However, relying solely on the chemical output of the Earth's crust meant that life was tethered to specific geological hotspots. Hydrogen was the limiting factor in primary production, a bottleneck that kept the early biosphere small and localized. To expand across the globe, life needed a more abundant and reliable energy source. The transition from eating chemicals to eating light was not a single, sudden event, but a series of evolutionary experiments. One of the most intriguing first attempts at harnessing solar energy is known as the Purple Earth hypothesis. Long before chlorophyll became the dominant pigment of the biosphere, early life may have relied on a much simpler molecule called retinol. Today, retinol is used by certain archaea, such as halobacterium, to capture light and pump protons across their membranes to generate energy. Unlike chlorophyll-based systems, which are complex and require a sophisticated electron transport chain, retinol-based phototrophy is relatively straightforward. Because retinol absorbs green light and reflects red and violet, these ancient organisms would have given the early Earth a distinct purple hue. This retinol-based strategy represented a fundamental shift. It was the first time life broke its total dependence on hydrothermal chemicals and began to tap into the vast energy flux of the sun. However, retinol-based systems had their limitations. While they were excellent at generating energy for cellular maintenance, they were not as effective at driving the complex carbon fixation needed for massive growth. This created an evolutionary opening for a second, more powerful light harvesting technology, chlorophyll-based photosynthesis. The development of chlorophyll and its bacterial cousin, bacteriochlorophyll, allowed life to move beyond simple energy scavenging and into the realm of true primary production. This transition likely began in the dim, infrared-rich environments of shallow hydrothermal vents or hot springs, where early pigments could capture the heat glow of the Earth before eventually adapting to the visible light of the sun. As life moved away from the vents and into the open ocean, it faced a new challenge. To fix carbon dioxide into biomass using light, microbes needed a steady supply of electrons. In the absence of the machinery required to split water, a feat that would not be mastered for another billion years, early phototrophs experimented with a variety of exotic electron donors. While hydrogen sulfide and ferrous iron were common, the early Earth was a laboratory of chemical diversity, where life also utilized arsenic, arsenite, and nitrite as exotic electron donors. The move to the sunlit surface was not without peril. Without an ozone layer, the Archean Earth was bathed in lethal doses of ultraviolet radiation. The first photosynthetic bacteria had to develop strategies to protect their delicate internal machinery. This led to the evolution of protective pigments and the habit of living in slightly deeper water or within the protective matrix of microbial mats, where the upper layers of cells could act as a biological shield for those below. This environmental pressure shaped the very architecture of the early biosphere, favoring organisms that could balance the need for light with the need for protection. The rise of anoxygenic photosynthesis also had a profound impact on the global nitrogen cycle. Nitrogen is an essential building block of life, required for the synthesis of proteins and genetic material. But atmospheric nitrogen gas is incredibly stable and difficult to break apart. In the nutrient-poor oceans of the Archean, bioavailable nitrogen was a scarce resource. Many of these ancient anoxygenic bacteria developed the ability to perform nitrogen fixation, the energy-intensive process of converting atmospheric nitrogen into ammonia. The ability to fuel this process with solar energy gave these organisms a massive competitive advantage. It allowed them to build biomass in environments where life had previously been impossible, effectively fertilizing the early oceans and setting the stage for a more robust and interconnected global ecosystem. The evolution of these light harvesting systems was not a simple linear progression from one ancestor to the next. Genomic analysis has shown that the components of the photosynthetic apparatus, the pigments, the reaction centers, and the carbon-fixing enzymes, often evolved independently. Through a process known as horizontal gene transfer, different groups of bacteria swapped these genetic blueprints like pieces of a modular kit. This genetic shuffling created a complex evolutionary tapestry, allowing various lineages to mix and match different light harvesting strategies to suit their specific environments. This modularity is a hallmark of microbial evolution and explains how such a wide variety of anoxygenic strategies could emerge and stabilize in a relatively short geological time frame. The success of these first light catchers fundamentally altered the trajectory of the planet, establishing these ancient microbes as the architects of the planet's first global ecosystems. This transition from a chemically fed world to a solar-powered one raised a new question. How would the Earth itself respond to this new, aggressive biological force? Chapter 2. The Iron Ledger. Geological Monuments of Ancient Light. To truly appreciate the power of anoxygenic photosynthesis, we must look beyond the microscopic cells themselves and examine the colossal monuments they left behind. These organisms were the planet's first great architects and alchemists, capable of taking invisible, dissolved minerals and transforming them into thousands of feet of solid rock. The geological record of the early Earth is not merely a collection of stones. It is a biological ledger, a preserved history of how ancient bacteria manipulated the chemistry of the entire ocean to satisfy their need for energy. The most spectacular evidence of this microbial alchemy is found in the banded iron formations, or BIFs, which represent some of the largest and most distinctive sedimentary deposits on our planet. In the ancient world, before the rise of oxygenic life, the oceans were ferruginous, saturated with dissolved ferrous iron that had been transported from deep-sea hydrothermal vents or weathered from the young continents. This iron stayed in solution, turning the oceans into a vast reservoir of potential energy. For a modern human looking at that ancient sea, the color would have shifted based on the state of the minerals within. A deep murky green where dissolved ferrous iron and dense bacterial blooms predominated, or a pale rust-like orange as iron oxides began to precipitate. This dissolved ferrous iron was the perfect electron donor for anoxygenic phototrophs. Geologists distinguish between two primary types of these formations, each telling a different story of the microbial environment. The Algoma-type BIFs are the older of the two, primarily Archean in age and closely associated with volcanic centers and island arcs. These rocks often display a europium anomaly, a specific chemical signature in the rare earth elements that confirms their origin from high-temperature hydrothermal fluids. These were the products of bacteria living in the immediate vicinity of the Earth's internal heat. In contrast, the Lake Superior-type BIFs, which appeared later in the Paleoproterozoic, formed on vast continental shelves. These reflect a shift in microbial scale, where bacteria began to process iron weathered from the continents themselves, sculpting the edges of the growing land masses. Scientific research into these ancient rocks, utilizing sophisticated isotope geochemistry, has provided a smoking gun for the activity of these bacteria. The Isua Greenstone Belt in Greenland, dating back 3.7 to 3.8 billion years, contains alternating layers of silver-gray magnetite or deep red hematite, sandwiched between layers of silica-rich chert. While geologists once debated whether these layers were formed by purely chemical processes, the emerging consensus points to the photoferrotroph. These anoxygenic bacteria lived in the sunlit upper layers of the iron-rich ocean. Using the energy of the sun, they stripped an electron from the dissolved ferrous iron, converting it into ferric iron. This oxidized iron was no longer soluble. It formed a fine rust-like gel that slowly drifted down to the ocean floor like biological snow. The sheer scale of these formations is difficult to wrap the mind around. In the Hammersley Range of Western Australia, these iron beds are over 3,000 feet thick and stretch for hundreds of miles. Similar deposits are found in the Iron Range of North America, the Carajás Formation in Brazil, and the Transvaal Basin in South Africa. These are the very same deposits that provide the vast majority of the iron ore used by human civilization today. Every steel skyscraper, every bridge, and every automobile is, in a very real sense, a byproduct of anoxygenic photosynthesis that occurred billions of years ago. These bacteria were effectively mining the ocean for electrons, moving more metal than any industrial operation in history. The banded nature of these rocks, the distinct, repeating layers, reveals a rhythmic pattern of life. Geologists have observed microbands that may represent annual or seasonal cycles, reflecting fluctuations in bacterial populations. During periods of high sunlight or nutrient availability, the photoferrotrophs would bloom, producing a thick layer of iron oxide. When conditions became less favorable, the iron production would slow, allowing a layer of silica or carbonate to settle instead. This cycle of boom and bust, written in stone, shows a biosphere that was already dynamic and responsive to its environment nearly 4 billion years ago. The first great era of banded iron formations came to a dramatic and potentially catastrophic end approximately 1.849 billion years ago. While the gradual rise of oxygen played a role, the geological record points to a sudden disruption, the Sudbury impact. A massive asteroid struck the Sudbury Basin in what is now Ontario, triggering a tsunami estimated to be 1,000 meters high. This colossal wave would have swept across the global ocean, violently mixing the stratified layers and oxygenating the deep ferruginous waters. This impact may have acted as a kill switch for the conditions that allowed BIFs to form, effectively ending the reign of the photoferrotrophs in the open sea. A critical tipping point in this transition is found in the appearance of manganese oxides in the rock record. Because the oxidation of manganese requires a much higher redox potential than the oxidation of iron, it serves as a definitive signal. When manganese oxides began to appear, it signaled that molecular oxygen had finally escaped the sinks of the ocean and was beginning to accumulate in the atmosphere. The geological record also reveals that the anoxygenic toolkit never truly went away. BIFs make a brief, dramatic reappearance much later in Earth's history, around 750 million years ago, during the snowball Earth events. During these global glaciations, thick ice sheets cut off the supply of atmospheric oxygen. The deep oceans once again became anoxic and ferruginous. When the ice finally melted and sunlight returned to the iron-saturated waters, anoxygenic photoferrotrophs staged a massive comeback. They bloomed on a global scale, creating the final, younger layers of banded iron formations seen in the Rapitan group in the Yukon and the Uricum deposits in Brazil. These ancient rocks force us to reconsider the definition of a productive biosphere. The BIFs represent billions of tons of biological waste product, suggesting that ancient anoxygenic populations were incredibly vast and active. They were the dominant force on the planet for 2 billion years. They took the raw materials of the Earth's interior, the iron from the core and the sulfur from the mantle, and used the power of a distant star to weave them into the crust of the planet. These ancient architects and alchemists established the biological legacy that would eventually set the stage for a billion-year stalemate. Chapter 3: The Billion-Year Stalemate. One of the most persistent mysteries in the history of our planet is not that oxygenic photosynthesis evolved, but that it took so long to win. For decades, the prevailing narrative was that once cyanobacteria developed the ability to split water and release oxygen, they possessed such an overwhelming competitive advantage that they must have rapidly dominated the globe. Yet, the chemical signatures in the rock record reveal a glaring paradox. Evidence for the existence of the oxygen-evolving complex appears as much as 1 billion years before the atmosphere finally became oxygenated during the Great Oxidation Event. For nearly 1/6 of Earth's entire history, the planet was locked in a biological and chemical stalemate. This was the Great Standoff, a billion-year competition where anoxygenic phototrophs successfully held back the rise of the modern world. The difficulty in pinning down the exact moment this standoff began is compounded by the Signor-Lipps effect. This paleontological principle reminds us that the fossil and chemical record is inherently incomplete. The first or last appearance of a signature in the strata rarely represents the true biological beginning or end of a process. Because the sedimentary record of the Archean is so fragmented, the billion-year gap might be even more complex than it appears, representing a series of fits and starts where oxygen rose locally only to be crushed by the surrounding environment. To understand how a group of primitive bacteria could thwart the expansion of oxygen-producing life for such an immense duration, we have to look at the ecophysiology of the ancient ocean. In the Archean and early Proterozoic, the sea was a highly stratified environment. At the surface, where sunlight was most intense, lived the early ancestors of cyanobacteria. Below them, in the dimmer, oxygen-poor depths of the photic zone, thrived the anoxygenic phototrophs. While the oxygenic organisms had the advantage of an infinite supply of water, the anoxygenic bacteria occupied a strategic position that allowed them to control the flow of the most precious resource in the sea, phosphorus. Phosphorus is the master switch of the biosphere, essential for DNA, RNA, and ATP. In the ancient ocean, it was a scarce and limiting nutrient, introduced primarily through upwelling from the deep sea. This created a vertical nutrient gauntlet. As phosphorus-rich waters rose toward the surface, they first had to pass through the layer occupied by anoxygenic phototrophs. Research into the model photoferrotroph chlorobium thioferrooxidans has revealed just how effective these organisms are at winning this battle. This bacterium possesses a highly sophisticated genetic toolkit known as the phosregulon, which includes a high-affinity phosphate-specific transport system. This system is so sensitive that the bacteria can continue to grow and photosynthesize even when dissolved phosphate levels drop to near zero concentrations. By sitting at the bottom of the photic zone and intercepting the upwelling phosphorus, anoxygenic phototrophs effectively starved the oxygenic populations living above them. This gauntlet effect meant that even though cyanobacteria had the superior electron donor in water, they lacked the raw materials to build large enough populations to oxygenate the atmosphere. The nitrogen cycle added another layer of complexity to this global stalemate. In the stratified seas of the Proterozoic, the boundary between the oxygenated surface and the anoxic deep, the redoxcline, was a site of massive nitrogen loss. As nitrate moved into the oxygen minimum zone, it was intercepted by microbes performing denitrification and anamox, processes that convert bioavailable nitrogen back into inert nitrogen gas. This created a nitrogen famine in the upper layers of the ocean. Anoxygenic phototrophs, however, often have a lower nitrogen requirement, or are better at fixing atmospheric nitrogen in anaerobic conditions. By consuming the remaining fixed nitrogen before it could reach the surface, they further limited the growth of the oxygen-producing cyanobacteria. The standoff was not just a battle for food, it was also a matter of chemical warfare. Many of the substances that anoxygenic bacteria depended on were actively toxic to the machinery of oxygenic photosynthesis. Hydrogen sulfide, the primary electron donor for many anoxygenic groups, is a potent inhibitor of photosystem II, the very complex that cyanobacteria use to split water. Scientific studies of the modern cyanobacterium Pseudonabiana strain FS39 provide a vivid window into how ancient life managed this conflict. This organism is a metabolic hybrid. It can perform oxygenic photosynthesis when sulfide levels are low, but as soon as it detects hydrogen sulfide, it shuts down its oxygen-producing system and switches to anoxygenic photosynthesis, using the sulfide as an electron donor. This flexibility suggests that for much of the early Earth's history, many oxygenic organisms were forced into an anoxygenic lifestyle by the chemistry of their surroundings. The transition between these two modes follows a strict kinetic regulation, where oxygenic photosynthesis only becomes active when the concentration of the preferred anoxygenic donor falls below a specific threshold. In the dim, nutrient-rich deeper layers of the ancient photic zone, the chemical environment would have almost always favored the anoxygenic mode, creating a self-reinforcing cycle that kept the environment anoxic. Beyond the ocean, the atmosphere itself was a major player in maintaining the stalemate. The standoff was reinforced by high rates of methane oxidation, which acted as a massive sink for any oxygen produced at the surface. Methane-producing microbes, or methanogens, thrived in the anoxic conditions of the early Earth, pumping out a potent greenhouse gas that also reacted greedily with oxygen. Scientific models of methane bistability suggest that the atmosphere was locked into a low oxygen steady state. Breaking out of this state required more than just biological effort. It required a shift in the planet's fundamental chemistry. One such shift was the nickel famine. Methanogens require nickel as a critical cofactor for the enzymes they use to produce methane. Research into ancient sedimentary rocks has shown that as the Earth's mantle cooled over hundreds of millions of years, the volcanic flux of nickel into the oceans began to decline. This nickel famine led to a crash in methanogen populations, which in turn reduced the atmospheric methane levels. With less methane to act as an oxygen sink, the cost of oxygenating the atmosphere dropped significantly, weakening one of the primary pillars of the Great Standoff. Simultaneously, a physical mechanism known as the photoperiod hypothesis may have been tilting the scales. In the Archean, the Earth rotated much faster, and days were significantly shorter, perhaps only 6 hours long. In these short days, cyanobacterial mats struggled to export oxygen. During the brief sunlit hours, the oxygen they produced was largely consumed by their own respiration or by the immediate chemical environment. As the moon's gravity gradually slowed the Earth's rotation, days became longer. These extended periods of sunlight allowed cyanobacterial mats to reach a tipping point, where oxygen production exceeded local consumption, allowing the gas to finally escape the mats and flood the surrounding water. The stability of this standoff is reflected in the long Middle Age of Earth's history, where the oceans remained redox stratified. The anoxygenic phototrophs were the masters of this intermediate state, utilizing wavelengths of light that oxygenic life could not see to occupy every possible niche where their preferred electron donors were present. Breaking the stalemate required a fundamental shift in the planet's geophysical plumbing. While sudden events like the Sudbury impact disrupted the stratified layers, the ultimate transition was driven by a runaway feedback loop. The growth of the continents increased the weathering of phosphorus-rich rocks, providing a nutrient surplus that allowed cyanobacteria to finally overcome the nutrient gauntlet. As oxygen levels began to rise, even slightly, a new feedback loop took hold. The rising oxygen reacted with the dissolved ferrous iron in the deep ocean, stripping the anoxygenic photoferrotrophs of their primary fuel. This sweeping effect caused photoferrotroph populations to crash, which reduced the competition for phosphorus and light. This allowed the oxygenic cyanobacteria to expand their range and produce even more oxygen, which in turn cleared more iron from the water. This transition was further accelerated by the formation of an ozone layer. As oxygen accumulated in the upper atmosphere, it formed ozone, which shielded the surface from ultraviolet radiation and fundamentally changed the rate of methane oxidation. This atmospheric transition allowed the planet to jump from the low oxygen steady state to a high oxygen one. The anoxygenic phototrophs, once the dominant gatekeepers of the global ecosystem, were forced to retreat into specialized extreme environments where oxygen could not reach. The billion-year struggle highlights that the history of life is not a simple march of progress, but a history of ecological power dynamics where the physical environment and the biological inhabitants are constantly negotiating the terms of existence. The specialized pigments and metabolic flexibility developed to win this standoff would become the enduring legacy of these ancient alchemists, allowing them to wait in the shadows for the world to change once again. Chapter 4. The Infrared Superpower, Mechanics of the Deep and Dark. To understand how anoxygenic phototrophs survive in the most inhospitable environments on Earth, we must look at the world through a different lens. While the plants and algae of the surface are locked into the narrow band of visible light, anoxygenic bacteria have mastered what can only be described as an infrared superpower. They have developed the art of living on the leftovers of the solar spectrum, thriving in the deep, the dark, and the murky shadows where visible light cannot reach. This capability is not a minor adaptation. It is a fundamental redesign of the photosynthesis machinery, centered around a unique class of pigments known as bacteriochlorophylls. The most familiar pigment in nature is chlorophyll A, the vibrant green molecule that powers plants. Chlorophyll A is highly effective at capturing blue and red light, but it is virtually blind to the long wavelength radiation beyond the red end of the spectrum. Anoxygenic bacteria utilize bacteriochlorophylls A through G, molecules with a structure specifically tuned to the near-infrared. In a laboratory setting, when these pigments are dissolved in a solvent like ether, they show absorption peaks around 775 to 790 nanometers. However, the true physics of the system manifests in the natural membrane milieu. When these pigments are embedded within the bacterial membrane, they undergo a dramatic redshift caused by extended resonance structures and specific protein-pigment interactions. In their natural environment, these bacteria can harness light at wavelengths as long as 1040 nanometers, light that is completely invisible to the human eye and unusable by any plant. The diversity of these pigments is a testament to billions of years of niche specialization. While bacteriochlorophylls A and B are common in purple bacteria, the green sulfur bacteria, GSB, utilize a specialized group of pigments known as bacterioviridins, which include bacteriochlorophylls C, D, and E. These allow GSB to capture light in the 720 to 755 nanometer range. Interestingly, science has identified a ghost pigment, bacteriochlorophyll F, which has been synthesized in the lab but has never been naturally observed in a living organism, representing a potential evolutionary path that nature, for reasons unknown, has not yet taken. This infrared sensitivity allows for a sophisticated vertical stratification in aquatic ecosystems. As light travels deeper into the water, high-energy blue and red photons are depleted by surface-dwelling organisms. Anoxygenic phototrophs have evolved to occupy the specific optical niches left behind, tuning their machinery to the green light and long-wavelength infrared that penetrate the depths. This allows different species to organize themselves by frequency, with each group harvesting the specific sliver of the spectrum that the organisms above have ignored. The structural housing for these pigments is as specialized as the pigments themselves. In the world of anoxygenic photosynthesis, there are no chloroplasts. Instead, the light-harvesting machinery is integrated directly into the bacterial cell architecture. Purple bacteria utilize intracytoplasmic membranes, extensive complex folds or spherical vesicles that branch inward from the plasma membrane. By creating these internal folds, the bacteria dramatically increase the surface area available for photosynthesis, packing their cells with thousands of reaction centers and light-harvesting complexes that function like internal solar panels. Green sulfur bacteria and certain members of the chloroflexodophilum employ one of the most efficient light-harvesting structures in the biological world, the chlorosome. These are giant ellipsoidal antenna complexes attached to the inner side of the cytoplasmic membrane. Unlike the pigment protein complexes in plants, where each pigment molecule is held in place by a protein scaffold, chlorosomes contain tens of thousands of bacteriochlorophyll molecules that are self-assembled into large organized aggregates. This massive concentration of pigment makes the chlorosome an incredibly sensitive light trap. It is so effective that green sulfur bacteria have been found thriving at the bottom of the Black Sea and near deep-sea hydrothermal vents, performing photosynthesis at light intensities once thought to be physically impossible for life to sustain. Most GSB are non-modal, but the family includes unique exceptions like chloroherpedin thalasseum, a gliding bacterium that can physically navigate the murky depths to find the optimal light and sulfide concentrations. The efficiency of this light capture is further enhanced by quantum walking. When a photon hits a pigment molecule, its energy is converted into a quasi-particle called an exciton. In the highly organized environment of a photosynthetic complex, the exciton behaves like a wave, effectively testing multiple pathways simultaneously to find the most direct route to the reaction center with near instantaneous speed. This ensures that almost no energy is lost as heat, which is crucial for organisms living on the absolute edge of darkness. Once the energy reaches the heart of the machinery, it enters the reaction center, where light is converted into chemical energy. In anoxygenic phototrophs, these centers come in two primary architectures, type 1 and type 2. While oxygenic life uses both in a linked chain, anoxygenic bacteria typically rely on only one, and that choice dictates their entire metabolic strategy. Type 1 reaction centers are found in green sulfur bacteria, heliobacteria, and certain acidobacteria. These are FS-type centers that use iron-sulfur clusters as terminal electron acceptors. A classic model is Chlorobaculum tepidum. In its reaction center, a special pair of bacteriochlorophyll molecules, designated P840, absorbs an exciton and enters a highly excited state. This makes P840 a powerful electron donor. It gives up an electron, which is passed through a series of internal carriers to ferrodixin. From there, the electrons enter a bifurcated chain. Some are used to reduce NAD+ to NADH for carbon fixation, while others contribute to the creation of a proton gradient for ATP synthesis. To reset the system, the electron-deficient P840 must be reduced back to its original state, a process mediated in GSB by a specialized protein called cytochrome C555. While type 1 centers are usually associated with anaerobic environments, nature provides a striking exception in Chloracidactyrium thermophilum. This unique aerobic phototrophic acid bacterium uses a type 1 reaction center despite living in oxygenated thermal springs, proving that the type 1 architecture is not strictly tethered to the anaerobic world. Type 2 reaction centers, found in purple bacteria and filamentous anoxygenic phototrophs, are pheophyton quinone centers. In purple bacteria, the process begins with a bacteriochlorophyll pair called P870. When excited, it donates an electron to bacteriopheophyton, a form of bacteriochlorophyll lacking the central magnesium ion. The electron moves through a series of quinones and eventually reaches a cytochrome BC1 complex, which pumps protons to drive ATP production. The type 2 system is primarily cyclic. The electron is often returned to the original reaction center by a soluble carrier like cytochrome C2. Because the electrons are recycled, these bacteria are incredibly efficient at producing ATP, but do not directly produce the reducing power, NADH, needed for carbon fixation. To solve this, they employ reverse electron flow, using the energy of their proton gradient to push electrons against their thermodynamic gradient onto NAD+. Recent metagenomic evidence has expanded our understanding of this architecture, suggesting that members of the Myxococcota phylum also possess photosynthesis gene clusters for type 2 reaction centers, a major phylogenetic expansion for a group previously thought to be non-photosynthetic. The heliobacteria represent another unique branch of this machinery. As the only known gram-positive photosynthetic bacteria, they lack the complex internal membranes of purple bacteria or the chlorosomes of GSB. Instead, their entire photosynthetic apparatus is located directly on the plasma membrane. They use a unique pigment called bacteriochlorophyll G. In a strange chemical bridge between worlds, bacteriochlorophyll G can be converted into a form of chlorophyll A when exposed to oxygen. Heliobacteria are primarily photoheterotrophs, using their streamlined system to power the uptake of organic acids in anaerobic soils. The modularity and mobility of these systems are best illustrated by recent discoveries in the seasonally anoxic waters of boreal shield lakes. These iron-rich, sulfate-poor environments serve as modern laboratories for the early Earth. Here, researchers have found populations of chlorobia that possess the genomic potential for photoferrotrophy, the ability to use dissolved ferrous iron as an electron donor. This process is linked to the CYC2 gene, which allows the bacteria to take electrons directly from iron. Even more remarkable is the discovery of chlorohelix allophototropa within these lakes. This spiraling filamentous bacterium belongs to the Chloroflexota phylum, a group that typically utilizes type 2 reaction centers. However, chlorohelix possesses a type 1 reaction center while still retaining the chlorosomes characteristic of its phylum. This transition form has revised our understanding of photosynthetic evolution, suggesting that reaction centers and light harvesting antennas are far more modular than one suspected. This serves as a further example of the photosynthetic genetic toolkit. The sheer variety of these strategies, from the massive chlorosome traps of the GSB to the cyclic loops of the purple bacteria and the iron-oxidizing machinery of the boreal shield, highlights the incredible adaptility of life. These organisms have mastered the physics of the exciton and the chemistry of the infrared to carve out a living in the deepest shadows of the biosphere. This mastery of the dim and the deep has turned them into essential players in the global ecosystem, but it also raises a profound question. How do these ancient systems, built for the dark and the anaerobic, negotiate the transition into the sunlit, oxygen-saturated world of the modern era? Chapter 5. The Modern Paradox: Scavengers, Symbionts, and Factories. The story of anoxygenic photosynthesis did not end with the oxygenation of the atmosphere billions of years ago. Instead, it shifted into a more complex and paradoxical phase. In the modern world, these ancient metabolic strategies have not only survived, but have diversified into roles that challenge our basic understanding of how life interacts with its environment. We find these organisms in the most unexpected places, from the sun-drenched surface of the open ocean to the toxic depths of industrial wastewater systems. They are no longer the undisputed rulers of the planet, but they remain essential ecological players, acting as master recyclers, symbiotic partners, and potential solutions to some of our most pressing environmental challenges. One of the most striking paradoxes in modern microbiology is the existence of aerobic anoxygenic phototrophs, or AAPs. For over a century, the scientific consensus held that anoxygenic photosynthesis was strictly confined to anaerobic, oxygen-free environments, as oxygen was viewed as a lethal inhibitor of these ancient pathways. However, the discovery of bacteria like Erythrobacter longus and Rhizobacter denitrificans fundamentally changed this view. These organisms are obligate aerobes, requiring oxygen to breathe, yet they also possess the machinery for anoxygenic photosynthesis. They thrive in the most oxygen-rich environment on the planet, the upper layers of the global ocean. In these vast marine deserts, where nutrients are vanishingly scarce, AAPs have carved out a unique niche. They can make up more than 10% of the total microbial population in certain oceanic regions. Unlike the cyanobacteria they live alongside, AAPs do not use light to fix carbon dioxide into new biomass through the traditional pathways. Instead, they use their light-harvesting machinery to generate a supplemental supply of ATP. This extra energy allows them to be more efficient with the limited organic matter they find in the water. It is a survival strategy that prioritizes energy conservation over total production. By using light to power their cellular maintenance, they can reserve their precious carbon for growth and reproduction. This hybrid lifestyle, part scavenger, part solar-powered, makes them one of the most successful groups in the marine biosphere. The internal regulation of these organisms is a marvel of biological engineering. Because they live in the presence of both light and oxygen, they are constantly at risk of producing singlet oxygen, a toxic byproduct. To manage this, bacteria like Rhizobacter shabai have developed a highly synchronized transcriptional response. They primarily synthesize their photosynthetic apparatus in the dark. When the sun rises, they immediately downregulate the genes responsible for making these light-harvesting complexes. This night shift production strategy ensures that they have the machinery ready to catch the first rays of dawn while minimizing the risk of self-inflicted chemical damage during the peak intensity of the day. Furthermore, Rhizobacter shabai demonstrates the metabolic diversity of the group by potentially utilizing the 3-hydroxypropionate cycle for carbon fixation, an alternative to the more common cycles found in other phototrophs. Moving from the open ocean to the coastal fringes, we find another fascinating dimension of modern anoxygenic life, the world of microbial symbiosis. The Rhizobiaceae family, famous for their role in fixing nitrogen in the root nodules of land plants, has a secret history rooted in anoxygenic photosynthesis. One of the most compelling paradox anecdotes in modern microbiology involves the genus Hoflia. For years, Hoflia were described in scientific literature as non-pigmented heterotrophs, simple scavengers that lived off the organic matter produced by others. It was only through recent genomic and physiological analysis that their true nature was revealed. They are functional anoxygenic phototrophs. Genera such as Hophlea are frequently found in close association with oxygenic phototrophs like diatoms, dinoflagellates, and cyanobacteria. For example, Hophlea phototrophica was isolated from marine dinoflagellates, suggesting a long-standing partnership where the anoxygenic bacteria live off the organic leavings of their oxygenic hosts, while providing essential services in return. This evolutionary trajectory is particularly evident in the genus Bradyrhizobium. Some modern strains of these bacteria still retain the ability to perform anoxygenic photosynthesis. In certain tropical legumes, these photosynthetic strains are high-performance partners. Studies have shown that photosynthetic Bradyrhizobium can nodulate their host plants without the need for traditional nod factors, and they demonstrate significantly higher rates of nitrogen fixation compared to their non-photosynthetic relatives. In certain unique geographical settings, we can still see the ancient stratified world of the Archean preserved in miniature. Meromictic lakes, characterized by water layers that do not mix, serve as living laboratories. In the Black Kitchier and Big Kitchier lakes of the Mare El Republic, a dense bacterial plate forms at the chemocline. Here, green sulfur bacteria like Chlorobaculum clathritiforme act as a biological barrier that intercepts upwelling hydrogen sulfide. In these systems, we also see complex symbiotic consortia, such as Chlorochromatium magnum, where a central motile bacterium is covered by anoxygenic phototrophs, allowing the entire colony to move through the water column to find the perfect balance of light and chemicals. Deep-water oligotrophic lakes, like Lake Foxian in China, offer another perspective on how these bacteria adapt. In these systems, researchers have identified Cyanobium, common Monodaceae, and Rosomonas as keystone taxa. There are distinct differences between free-living bacteria and those attached to organic particles. The free-living fraction is often enriched with bacteria capable of aerobic anoxygenic photosynthesis, while the particle-attached communities focus on the decomposition of organic matter and nitrogen metabolism. This spatial separation shows that anoxygenic strategies are part of a broader ecological division of labor that keeps these nutrient-poor ecosystems functioning. The persistence of these organisms in nutrient-starved environments is driven by extreme physiological adaptations. While the nutrient gauntlet of the ancient oceans was a battle for survival, modern strains have refined this into a high-tech art form, utilizing the high-affinity phosphate transport system, Phoregulon, of Chlorobium phaeoferroxidans. The unique metabolic talents of anoxygenic phototrophs are now being harnessed for industrial and environmental biotechnology. One of the most promising applications is the detoxification of hydrogen sulfide, a frequent and dangerous contaminant in municipal and industrial wastewater, as well as in natural gas and biogas production. Anoxygenic sulfur bacteria offer a biological alternative to expensive chemical treatments. The industrial utility of these bacteria is dictated by their specific biological habits. Because green sulfur bacteria, GSB, deposit their sulfur granules extracellularly, outside the cell wall, they are the preferred choice for industrial recovery. The resulting elemental sulfur is insoluble in water and easily separated, allowing for the recovery of a valuable raw material for fertilizers and chemicals. The versatility of purple non-sulfur bacteria, such as Rhodobacter spheroides and Rhodopseudomonas palustris, has made them stars of the bioremediation world. These organisms are among the most metabolically diverse life forms known to science, capable of growing photoautotrophically, photoheterotrophically, or through aerobic and anaerobic respiration. This flexibility allows them to thrive in the fluctuating conditions of wastewater treatment ponds. They are particularly effective at reducing the chemical oxygen demand of industrial runoff, breaking down complex organic pollutants. In the process, they produce a high-value biomass rich in proteins, vitamins, and antioxidants, which can be repurposed as animal feed. These bacteria are also being explored as microbial factories for the production of sustainable materials. Under certain conditions, many anoxygenic phototropes accumulate large quantities of polybeta-hydroxyalkanoates, PHAs, natural polymers that serve as energy storage. Because PHAs are fully biodegradable and can be produced using solar energy and waste products, they represent a sustainable alternative to petroleum-based plastics. Similarly, species like Chlorobium limicola are being studied for their ability to accumulate significant reserves of glycogen during photosynthesis, offering a potential pathway for the production of bio-based chemicals and fuels. The quest for renewable energy has also turned its attention to the hydrogen-producing capabilities of these bacteria. During the process of nitrogen fixation or through specialized hydrogenase enzymes, many anoxygenic phototrophs release molecular hydrogen as a byproduct. Unlike the hydrogen produced by industrial steam reforming, biohydrogen produced by bacteria is a carbon-neutral energy carrier. Researchers are currently working on optimizing photofermentative methods using purple non-sulfur bacteria to convert agricultural and food waste into clean-burning hydrogen gas. The study of these modern phototrophs has required the development of specialized tools to distinguish their activity from that of plants and algae. In environments like sewage ponds or microbial mats, oxygenic and anoxygenic organisms are often intimately mixed. Pulse amplitude modulation, PAM, fluorometry has emerged as a key technique for measuring these overlapping processes. By using specific wavelengths of light to excite different pigments and employing inhibitors like diuron to temporarily block oxygenic photosynthesis, scientists can estimate the electron transport rates of each group independently. This allows for a precise quantification of how much anoxygenic work is being done in a given ecosystem, revealing that these bacteria are often contributing far more to primary production than was previously realized. Even in the most extreme modern environments, anoxygenic photosynthesis continues to push the boundaries of what we consider possible for life. In toxic gold mine drainage systems and near hydrothermal vents on the ocean floor, these bacteria are found using the dimmest glows to survive. The persistence of ancient metabolic legacies in modern toxic niches is further evidenced by the use of arsenic, arsenite, or nitrite as exotic electron donors in anoxygenic photosynthesis. As we explore the potential of these organisms in environmental management and resource recovery, we are essentially learning to speak the chemical language that these bacteria have used for billions of years. We are finding that the very traits that allowed them to survive the harsh conditions of the early Earth, their ability to thrive in the dark, to process toxic chemicals, and to coexist in complex communities, are exactly the tools we need to build a more sustainable and resilient future. The paradox of the modern phototroph is that an ancient process has become one of our most sophisticated allies. The vertical stratification that began in the Archean oceans now finds a new home in the organized layers of a bioreactor, where the alchemy of light and sulfur continues to transform the world. As we look toward a future of bio-based manufacturing and carbon-neutral energy, one must wonder, what other metabolic secrets are still hidden in the genomes of the non-pigmented scavengers of the deep? From the murky, iron-saturated oceans of the Archean to the high-tech bioreactors of today, anoxygenic photosynthesis remains a silent architect of our world. These ancient alchemists did more than survive the early Earth. They sculpted its geology through banded iron formations and held the rise of oxygen at bay for a billion-year stalemate. By mastering the infrared spectrum and thriving in the deepest shadows, they proved that life's ingenuity is not limited to the visible light of a green leaf. Today, their unique metabolic toolkits offer us a path towards sustainable fuels and a deeper understanding of biological resilience. As we search for life on distant alien worlds, we must remember that Earth's own history was written by those who first found power in the dark. Thank you for listening.